Okay, now that we've learned how these microbes grow, now we have to learn how to stop these guys. And this is really, really important in medicine, in healthcare, because we have these organisms everywhere. We're going to talk about these organisms that cause nosocomial infections, those infections you get when you go to the hospital, all right, or a healthcare facility because they're out there. All right. And so it's important that we learn how to control these organisms. You will have a department in a hospital that actually works on that. There are cultures that are done on a regular basis to just basically test the different units in the hospital to see if there's underlying organisms. All right. So first of all, is some terms you need to learn, and you need to know these terms. Okay, so I'm telling you right now, these terms will be on the exam. All right, so you need to know the difference between an antiseptic, an anicidal, a degermer, what sanitation is, what sterilization is. So there is a difference in each one of these. So I've got this chart. I know it's from a long time ago, but it works, and it's the best chart I could find. And this helps you. Okay, and we'll go through some of these. All right. So first of all, we have to look at killing. All right. What we want to look at is when we try and evaluate these methods of microbial control, we need to look at the killing time. All right. And the killing time is actually, a lot of it is printed on the stuff, not necessarily saying killing time, but what you want to look at is the percentage of the population that is killed each minute. That determines how long you have to put this particular killing agent on the surface or on the body. So like if you come into the lab, we're told, every so many hours we spray our benches down and we let them sit for 15 minutes to make sure that all these lovely little organisms we could get from the blood are killed on the countertops. The same thing. You don't just, spray it with alcohol, and walk away. You've got to allow these organisms, the time for this agent to kill these organisms, okay? So just FYI. Okay, so when we look at the actions, there's different actions of how these antimicrobial agents work, okay? They can alter the cell walls or the membranes of these organisms, okay? The cell wall, if it has one, depends on, again, we talked about it, some organisms don't have cell walls, but if it has a cell wall, if you disrupt that cell wall, that kills the organism. The cell wall is what protects the organism from rupturing when you put it into different environments. You take that cell wall off, just like our blood cells, you put it in a water environment, it's bursted, it's done, okay? So, it's, if you can burst the cell wall of something, depending on what it is, that's a pretty good thing. You can work on the membrane, because if you damage the membrane, the cellular contents leak out. That kills the cell. When we get to viruses, some viruses are enveloped. And we'll talk about that when we get to viruses, but there's an envelope around that virus. An enveloped virus is much easier to kill than a non-enveloped virus because if you damage the envelope, that takes care of the virus. Okay, so just FYI. All right, so let's look at some of these. The other thing we can do is we can't damage the cell wall. We can't damage the outside of this organism. Maybe we can damage something inside the organism. So we can do damage to the proteins and the nucleic acids so it can't reproduce. There are some antimicrobial agents that will change the 3D shape of that protein. Most of you already know you expose it to heat or to certain chemicals that will quote unquote denature the proteins, i.e., take that protein apart, ruin the bonds between them, and therefore proteins have to be folded in particular shapes to work. Okay. I may or may not have had biochemistry, but proteins have a structure. And one of the structures that we talk about in forming a protein is what's called the quaternary structure, which is you get the sequence of the structure, but then in order for that protein to work, it's got to fold properly. If you have hemoglobin, it is supposed to store iron. It's got to fold properly in order to have that iron molecule in there. So if you can unwind or adjust that protein structure, that actual folding structure, then that protein can't work, okay, or doesn't work as well. So we can use extreme heat or certain chemicals to do that. You can also use the heat to destroy the nucleic acids. It can produce mutations that are fatal to the organism. It can halt the protein synthesis. Again, if you get it too hot, these things don't work. Okay? So when we look at this, there's some of the ways. So what do we want when we start to choose? I mean, you go into a hospital and they have certain chemicals that they're using, and you use some in your own home. But how do we determine? Okay, how does the epidemiology department of the hospital determine what agents are going to be used? Okay, so if you have an ideal agent, you want it first of all to be fairly expensive. You don't want it to cost a lot. You want it to be fast acting. You don't want to have to wait 4 hours for it to take effect. You need it to stay stable during storage. I mean, some of these things can become unstable and become dangerous if they're stored too long. But mainly you want to be capable of controlling the growth of these microbes that you're trying to kill. But you want to control the growth of the organisms that are harming you, but do not harm you, the objects that you may be putting it on, animals, that sort of thing. So you want it to work against the microbe that you're trying to kill, but not harm you. Okay, those are some of the really good qualities of what we would say is an ideal agent. Okay, so the other thing we have to think about is, where are we treating this? Okay, you can't put a harsh chemical or extreme heat on humans, on animals, on fragile objects, so you can't use those harsh chemicals. And I mean, you have to be very careful if you're doing a medical procedure. You do not want to damage tissue, right? But if you're cutting somebody open in an OR, you need those microbes down, because if you don't have that level of the microbes down, you get a post-op infection. So there's certain areas of the hospitals that need to have tighter controls versus some of the others, and that's one of the things that we have to think about. All right. So when we look at this, we want to look at these organisms, and this is basically, some organisms are very difficult to kill, and some of them are easy to kill. So here's the list from the most resistant to the most susceptible. And as I said, these envelope viruses, once you take the envelope off, they're gone, they're done. The viruses are not a living organism. So once you get that envelope off, that virus is killed. And you'll go up here, you can see these gram-positive bacteria, your staph and your streps are generally fairly easy to kill, except for those that are drug resistant, but and begin to develop these resistances to some of these chemicals. But in general, gram positives are easier to kill than gram negatives. And you can see then they're non-envelope viruses, and then your fungi. Gram negatives can be a little bit difficult to kill. We'll get into protozoans. Those are a little hard to kill. What we call Mycobacterium pneumoniae. Here we go. And here's the tough guys. Okay? We talked about endospores. So bacteria with an endospore is hard to kill. And you've got an experiment coming up in a couple of weeks that we will actually test this, because we'll be able to be testing the use of ultraviolet light on the bacterial, and you'll get to see the difference between having an endospore and not having an endospore. Prions are just an effective piece of protein, so this is going to be very difficult to kill. Prion diseases like mad cow disease, that you have to kill the herd. You have to kill the cow. We don't have a way right now. They're developing things, but we don't really have a way to kill the prions. That's a really, really tough thing. Okay, so we go from really fairly easy to kill to almost impossible. Okay. All right, some of the things that can affect the efficacy or effectiveness of these methods. First of all, again, what I just talked about, the susceptibility of the microorganism itself. How easy is it to kill? And so when we use these control methods, we classify particularly germicides as either high level, intermediate level, or low level. So your low levels will kill those organisms at the bottom of the list. The intermediate ones will kill the middle of the list. And the high levels will kill all the pathogens, including the endospores, but not the prions, unless you incinerate them. Okay? So when you look at it, we have different ways of doing this. We have physical, chemical controls. First of all, we're going to go over the physical methods of doing this. And we can expose them to extremes of heat, extremes of cold. We can take the moisture out or desiccate them. We can filter them using very, very, very tiny little pores. We can use a change in osmotic pressure, water, or we can irradiate them. Okay, so let's take a look at doing this by heat. This is probably, other than spraying things down, one of the most common things that we do in a hospital setting or doctor's office setting, and that is using moist heat. Okay, and we can disinfect, we can sanitize, or we can sterilize. So go back to those definitions about what that meant. Moist heat kills by, we denature the proteins. We break up that quaternary structure of the protein, and it's just non-functional. It will also destroy the membranes. It's more effective than dry heat because the water is a better conductor of heat than air. So it's more effective in getting that heat into the organisms. Now, some of the things that we use commonly in the hospital or in a lab is, and we use it in our homes, we boil. Put something in boiling water. It works out real well. Common instrument in a doctor's office and in hospitals is an autoclave, and we'll take a look at that. And that is just high heat at high pressure, and it will sterilize. Okay, and you will actually do put in a little control that tells you, yes, I have this is sterile now, because we'll use a strip that's impregnated with these organisms that withstand high heat. And if they die, then we know the autoclaving has worked. We can use pasteurization. That's used. It's a little slower, a little lower. We'll talk about that in a second. We're going to go through this. But pasteurization, pasture. You get pasteurized milk. Okay, and then we can do ultra high temperature sterilization. That's more usually used in not necessarily hospitals, but in industrial type of things and food preparation, when companies are making food. All right, so for boiling, you need to kill the cells of the bacteria, the fungi, and most viruses 10 minutes at sea level. If you're above sea level, you need to boil it for a little longer. Just FYI, if you're up in, the mountains of out west, up in Colorado, it takes a lot longer to boil. I'm telling you that right now. Okay. Temperature cannot exceed 100 degrees Celsius at sea level. It's impossible because that's what it boils at. So steam will carry some of this heat away. The boiling time is critical. And again, water boils at a lower temperature at the higher elevations. So it does require a longer boiling time because your pressure is down. There are some endospores and some protozoal cysts. Some viruses can survive boiling. But most of them that are going to have problems with us will not. Okay, because they got to survive in our body. We're not at that temperature. Okay. All right, autoclaving, as I mentioned, this is a pressure applied to boiling water, and it's encased so the steam cannot be released, and you get it up to boiling temperature. The pressure increases, and at 121 degrees Celsius, 15 psi for 15 minutes. Will kill anything. It will sterilize. Okay. And usually what you have in a doctor's office is a small desktop autoclave. At the school, we have desktop autoclaves a little bit bigger. In the hospitals, you have big autoclaves for surgical instruments and that kind of stuff. Okay. And there's your autoclave chart that gives you the temperature and the pressure, and you can see it. Okay. Pasteurization. This is made famous by Louis Pasteur, who discovered this. And today, again, in milk products, we use it, fruit juices. I do recommend that check. There's a lot of people drink raw milk. You can get diseases from raw milk, just FYI. You have to be careful with raw milk. But we use it today for our typical milk products. It is not a sterilization. So your very heat tolerant and heat loving microbes will survive. Basically, these do not cause spoilage. So they are generally not pathogenic, and we generally don't have to worry about them. Now you can see the 3 methods of pasteurizing milk. Yeah. All right, I put the Fahrenheit out there so that you have an idea. But yeah, it doesn't take long to pasteurize this milk. All right, now we can use high heat, which is ultra high sterilization. Yeah, 284 degrees Fahrenheit for 1 second and then rapidly cool. And you can treat these liquids can be stored at room temperature. So usually these are some kinds that are used in food preparation and stuff like that. Not something we use at a hospital or healthcare facility. We can use dry heat. This is used when we can't sterilize something with moist heat. Maybe we have something that can be damaged with the moist heat. And this dry heat can denature the proteins and that sort of thing. It requires higher temperature for a longer time because again, the water gets in better with the moist. Dry heat is also incineration, which is our ultimate means of sterilization. We get rid of everything. But, dry heat is not something we usually use, but You use it when you bake. Think about when you put stuff in the oven, well, you're using dry heat, right? We can use refrigeration or we can freeze things. This will decrease the metabolic rate of these organisms. It will decrease the growth rate, the reproductive rate. Chemical reactions will slow down again because the proteins are going to get stiff. Liquid water is not available if you put it below freezing. And the problem is we do have these organisms, psychrophilic organisms that like these cooler temperatures, like Pseudomonas is notorious in some of these molds, that they can multiply in refrigerated foods. I mean, I know you guys have seen plenty of times, you go in, you reach in the refrigerator and cheese has got mold on it or whatever. Yeah, those are these organisms that can survive, okay? So it does halt the growth of most pathogens, but organisms can grow. Slow freezing is more effective than quick freezing because of the way it affects the water in the cells. And organisms will vary in their susceptibility to freezing. You have to be aware of the organisms. But this is not something you guys generally have to worry about. All right, we can desiccate. We can basically take the water out. This is used frequently. It inhibits the growth. It's only microbiostatic, all right? So basically, you can use this. It's not something we generally do. Something that's frequently seen in the hospitals are things that are lyophilized. A lot of times your vaccines will come out lyophilized. A lot of the stuff we use in the lab is lyophilized. In other words, it's had the liquid removed, and basically you have a little vial with powder in the bottom. And then in order to use it, you have to reconstitute it. You have to add sterile water to that and mix it, and then it's viable, okay? But lyophilization is used for long-term preservation of microbiology cultures. Frequently in the lab, I'll get in cultures. If I'm doing a lab micro lab, I get a culture and I get a little vial and it's full of a microbe. And I just reconstitute it, put it out on a plate, put that plate in the incubator, let it grow, and I've got that organism to work with for class, okay? So works very, very, very well. And again, we also do it with vaccines. We can use osmotic pressure. I know you're familiar with this because any really salty food, like pickling your foods, are in high sugar. I mean, think of honey. Honey doesn't spoil. Why? It has a high concentration of sugar. Same thing if you do your pickles. I mean, you may get something on the top, but everything underneath there is fine. So high concentrations of salt or sugar will inhibit the growth. And cells in a hypertonic solution of salt or sugar, the concentration of hypertonic solution means the concentration is higher outside than it is inside the cell. Water's going to move out of the cell, and the cell desiccates. The water gets removed. Fungi have a greater ability to survive this than bacteria, and particularly they can survive in hypertonic solutions. You put a fungi in hypertonic solution and in a salt, they can survive pretty well. Okay. Another thing that we can do is we can use radiation. Wavelengths that are shorter than 1 nanometer, X-rays, gamma rays, and they basically eject electrons from the atoms to create ions. And what that does is it denatures, like DNA, it creates hydroxide bonds and that's ions and that sort of thing. You can use electron beams. They don't penetrate walls. Gamma rays, but they require hours. This is not something we usually use in a hospital. There are some things that we do. X-rays require too much time to be practical. You can irradiate food. Europe, there's controversy on irradiation, but a lot of foods are irradiated to prevent spoilage. And if they are, you'll see this symbol. It tells you it's been irradiated, and it doesn't hurt you, but it does help preserve the foods. Okay. Now, one of the things we do use a lot is non-ionizing radiation. We use this in a lab. You'll see an experiment done on this. And basically what this is ionizing radiation is shorter than 1 nanometer. The non-ionizing is greater than 1 nanometer. And what it does, it affects that 3D structure of the protein again and the nucleic acids. And it causes what's called thymine dimers in DNA. And basically when you're looking at your sequence of DNA, anytime you have the thymine next to the thymine, they're going to bond together rather than, I have another little thing in there, PowerPoint later on radiation, that sort of thing. And the one that I've got, when you get to the lab, the UV light on bacteria, which is in a couple of weeks, I'll show you what that thymine dimer looks like. But it works very well on surfaces or transparent fluids or air. Okay, it doesn't penetrate well. So there's an experiment that you'll see again in this lab where you put just a simple index card over half of the agar, and that little piece of paper is enough to block the UV light. You can't, even the lid of the agar plate, it won't penetrate. So you can't have anything between the UV light coming down and the surface you're trying to sterilize. It is very suitable for disinfecting air. A lot of hospitals use it in the ORs or doctor's offices and stuff like that. I've got some pictures of rooms because it's very effective at disinfecting the air. All right. If we want to disinfect a particular fluid, a transparent fluid, a surface, OR table, it's very effective. Okay. So just FYI. And again, we use it frequently in the hospital. Okay. Again, this irradiation of food destroys, dangerous pathogens such as salmonella, E. coli, listeria in raw meat and poultry and seafood. It delays the ripening of the fruit, stops the root vegetables from sprouting, extends overall life. It actually kills insects and pests that may be in spices. Frequently, your spices are irradiated because it will kill these little critters so that you don't have to use a pesticide or a fumigant or something like that. So irradiation, it can be very positive. Okay, then we can use chemical means. And here we're going to affect several pieces of that organism. And the effect varies with the temperature, how long it's exposed. The main thing here is how much organic matter. And again, this also applies to when you try and sterilize something by using an autoclave. You have to wash these things first. You have to wash these instruments first and then put them in the autoclave clean. Because if you have organic matter like tissue, welcome to your ORs, it's not going to work. So you have to make sure that any tissue has been removed, any organic matter. These The effectiveness will vary with the concentration, particularly the age of the chemical. I mean, it's been sitting around for 6 months, or is this, fairly brand new? Tends to be more effective against our lovely envelope viruses, okay, and up to the protozoans. But they do work pretty well. And these are things, we're going to go through these, that we use frequently all the time in the hospitals, okay? And again, this is what, when you have infection, your infection control people will really look at this and determine what is the best thing to use in the patient's rooms, in the labs, because you need different kinds of things that would be used, but it's up to them to determine that. Okay. So, first of all, we have phenols. These are, and again, intermediate to low-level disinfectants. They will denature, they will disrupt the cell membrane. They are effective in the presence of organic matter. Ooh, this is a good one. And they can remain active for a long time. They are commonly used in the healthcare setting, in the labs, in the homes, and the one you probably heard of is Lysol. Very good. Triclosan is also another one. The main problem with these is they have some side effects. People kind of choke on it, but it doesn't smell real good when you spray everything with Lysol. Most people, they try and hide it with scents, but phenols are usually pretty strong in smelling. There are some other ones that the hospital uses. Alcohol. This is an intermediate disinfectant. Okay, it evaporates rapidly. So this could be advantageous or disadvantageous. So you want it to, evaporate rapidly so that you can stick the patient, or do you need to let it sit? Okay, I know in a hospital, in our laboratories, we spray them down with alcohol or whatever, and we let it sit 15 minutes before we touch that countertop. Okay. Example is, swabbing your skin with 70% alcohol, ethanol prior to an injection or getting blood drawn or something like this. So it's a very good agent. It works rapidly. It dries fairly rapidly, and it doesn't take everything off. Again, it's a disinfectant. Okay, so it's going to lower the microbe level, but it's not going to totally take everything away. Okay, so just FYI. All right, aldehydes. This is a compound that ends in, CHO, and it will cross-link with a bunch of other things in these membranes and proteins to inactivate them. Like they'll interact with the sulfur molecules in there, the hydroxyl ions. One of the more famous ones is glutaraldehyde, and it can do short-term or long-term exposure. Glutaraldehyde is used a lot of times to sterilize or disinfect. It depends on how long it is. These instruments that cannot go into an autoclave. For example, some of these scopes, they have rubber and plastic pieces. You can't put a plastic scope or something that has a rubber piece on it in an autoclave. It doesn't work. Okay. So what they will do is they will soak it in glutaraldehyde overnight. The one thing with glutaraldehyde is that you don't have to change it every single time, but you do need to change it frequently. I've seen some docs that do not change this glutaraldehyde frequently enough, and you can get infections from it. So if you work in an office where you are using glutaraldehyde, make sure they change it if they have these kind of instruments where they have to, basically disinfect so they can use it. All right. Formalin, formaldehyde, same thing. It is used in disinfection of instruments. It is used in embalming. That's how you're embalmed. It is used, I can tell you, in the histology lab, tissues get sent down from surgery or whatever, and it is placed in formalin, and basically a block is made and it's sliced, and that's how the pathologist can look at the tissues. And those slides and those tissues that are encased in that formalin can stay for practically ever. Okay, but you don't really want to disinfect with it too much because formaldehyde is a carcinogen. I will tell you that. People that work in the histology labs have to wear the little devices that detect whether they have been overexposed to the formaldehyde. So it's not something we really want to use to disinfect a room or an instrument unless we absolutely have to. Okay. All right. Then we have halogens. These are the guys, if you've had any chemistry, are in that a certain line in that lovely little periodic table. Not the very last one, but not the very last one on the right-hand side, but the next column to it. Okay. And so these are intermediate level chemicals. This is your iodine, your chlorine, your fluorine, your bromides. They're pretty good. So we use iodine. You can do the iodine wipes. A lot of times for surgery, instead of using alcohol, they're going to, take that surgical area and wash it down with an iodine, betadine, iota-4. We treat water with chlorine. We use bleach. You can use bromine to disinfect hot tubs. So these halogens are really, really, really good because nothing really kind of interacts with them. Oxidizing agents. These will have O on the end of their chemical formula, but we have peroxides, ozone, periodic acid, and they're going to oxidize the enzymes. They're just going to break these enzymes up. So this is a high-level disinfectant or antiseptic. Hydrogen peroxide can disinfect and sterilize the surface of objects, but you don't. People like to pour, hydrogen peroxide on an open wound. It's not really good for doing that. I know people think about that, and it does not, but hydrogen peroxide basically catalase neutralizes it. So any organism like staph that has catalase and catalase positive will neutralize the hydrogen peroxide, and it can also damage the tissues that you're pouring it into. So you could use something else better than hydrogen peroxide, but if you're just trying to sterilize the surface or something, hydrogen peroxide works real well. We do use ozone treatment of drinking water. Peracetic acid is real good at killing spores, and it can be used to sterilize equipment. Not very often, but it can be. We can use enzymes. This is something that we're looking more into. They act against these organisms. We have natural enzymes, and I mentioned lysozyme in our tears, which digest the peptidoglycan wall of the bacteria. You all know about that now. So lysozyme is also used to reduce the number of bacteria in cheese. There is a prion that can remove the prions on medical instruments. So that we're moving more into using enzymatic action on some of these things. Just FYI. Surfactants. Surfactant is something that basically reduces the surface tension of something. So like if you wet your hands and you put your hands together and you try to pull them apart, it's harder to pull them apart because the water has this surface tension. Surface tension is what allows a little insect to walk over the top of water. Okay, so if you can reduce that surface tension on the water or that solvent, it makes it more effective. Some of the things that help is soap. That's what makes soap so effective, is that it reduces the surface tension of water and allows the soap to get in there. Okay. And so it allows you to degerm things, not disinfect, but degerm. All right, get the major pieces of those germs off and going there. Okay. We'll talk about quartz. It's in the, I'm going to skip that. They're used for medical instruments at a low level disinfectant. And I've got an explanation of quartz in the lab coming up, so I wouldn't worry about it right now. Heavy metals. These have been used frequently. Basically, they're low-level bacteriostatic fungicide. Babies, when they're born, 1% silver nitrate drops will be placed in their eyes to prevent blindness. They just do it because it will stop the Neisseria gonorrhea. All right. We used to use thimerosal frequently to preserve vaccines. We have gotten away from it because thimerosal is mercury. You don't want too much mercury in your system, but it can be used to preserve some vaccines that are really hard to work with. Copper is frequently used, although in fish tanks and swimming pools and stuff like that, interferes with chlorophyll. Copper, anytime you use a heavy metal, you have to worry about heavy metal poisoning. So you got to be careful about what you're using it for. Okay. A lot of swimming pools, they've gone to chlorine more than using basically your copper because people are swimming in the pools and swallowing water. I've seen you can put it in a fish tank and it's very effective, but you don't want to put it in an area where you're going to be eating the fish. Okay, so you just have to think about that because copper is a heavy metal and you can get heavy metal poisoning. Okay, and then we can move to gaseous agents. You can use the gas. I will tell you, I had one hospital that this is the way they used to sterilize the instruments for the OR. Basically, they can be used in hospitals, doctor's offices, dentist offices, not necessarily too many, doctor offices, but in hospitals, when you've got to sterilize large amounts, you can pump ethylene oxide in. It will denature, sterilize fairly quickly, but it can be very hazardous. You have to be very careful. I mean, I know in the hospital that I worked at, again, we used the ethylene oxide to sterilize the instruments because we had a very busy OR. And if you have a leak, you have about 8 seconds to get out of the room, and you're told if you walk in there, you hear this alarm go off, get out. Because once the door will close in so many seconds, and if you're not out, you're going to get locked in. So it's highly explosive. It is poisonous. It is carcinogenic. But the main thing is it takes the oxygen out of the air. And so, yeah, it works real well, but it can be dangerous. But, when you're trying to sterilize large amounts of instruments and stuff, it can be very effective. Okay, you can use antimicrobials. All right, we use these. And we'll talk about antibiotics. We'll have a whole lecture on antimicrobials a little later on. But the antibiotics, you have semi-synthetic and synthetic chemicals that are used to treat disease. Some are used for antimicrobial control outside the body. So you can use these antimicrobials in certain instances. All right. So then we go to mechanical ways. We finished the chemicals, now we can go into the mechanical means. And this is where we physically can get these bacteria, these microbes, out of liquids or gases. And when you have these heat sensitive solutions like pharmaceuticals, where you're making certain cough syrups or whatever it may be that you need to drink, or you need to create something like that in a pharmaceutical, you can use it. You can use HEPA filters in the hospitals and the labs to create a sterile environment. You can use positive pressure. I mean, you use them. When you have a patient that has tuberculosis, you don't want people to catch that. So you have to have special filters in those rooms to prevent the spread. One thing that we do use in the labs, and this is standard equipment in a laboratory, is a biosafety cabinet. Sometimes you'll hear it, we're working under the hood. That's what they mean. So this is a biosafety cabinet. If you came to campus and worked in the lab, you would see one. And basically, you have an airflow. Air comes in, and there's an exhaust fan out, and you have a glass screen, and the air, flows like this. You're working underneath this. And this glass, if you lift it too high, an alarm will ring and say, you got your glass too high, you need to lower it down. I used this when I was doing stem cell transplants, and I had to take that collection of stem cells and process those stem cells and then freeze them. And as I'm processing them, I didn't want any contamination of those cells because these patients have no bone marrow, basically. So you don't want to give them a disease. So we have to be very careful in working with those stem cell transplants. And this is one way we can do it. Okay, this is very commonly done. Okay. We'll talk also, one of the problems that we run into because of we're doing this is that resistance. There is little evidence that extensive use of the products adds to human, animal, or health in terms of this. What we do want to do is we use these products to promote an environment where we've lessened the load, the bacterial load. Okay. The use of these products can promote the development of resistant microbes, particularly antibiotics. You can get organisms that develop the resistance to certain of these chemical means. High heat and sterilization, no, but other than that, some chemical mean or something like that, they can develop resistance to it. Okay. So just FYI, think about it. We're going to have a whole lecture about how these organisms develop resistance. And we're going to talk about these. So FYI. All right, so hopefully you've learned a little bit about this. And this is, again, just a beginning. We'll talk a little later on some of this other stuff. When you use these things, what they recommend is that we limit the use of these things to just food handling and health care involving, high-risk patients and newborns, and try not to do too much because we're developing a lot of resistance with these organisms, and that's not something, we've already got enough as it is. But again, you're going to have a lecture on the resistance of these guys. All right. Okay.